In classical biology, reverse transcriptases follow a strict rule. They must read an existing RNA strand to synthesize a complementary DNA strand. Bacteria and the viruses that infect them, bacteriophages, exist in a continuous evolutionary arms race. This pressure forces bacteria to develop increasingly extreme countermeasures. One of the most severe responses is abortive infection, or ABI. When a bacterial cell detects a viral breach, it initiates a localized scorched earth protocol, killing itself to prevent the virus from replicating. The executors of this cellular shutdown are defense-associated reverse transcriptases, known as DRTs. Today, we are looking specifically at the mechanism of DRT7. DRT7 does not block the initial viral attachment to the cell membrane. Instead, it waits to halt the infection at a post-adsorption stage, deliberately trapping the multiplying viral progeny inside the host. To execute this defense, DRT7 relies on two distinct structural regions working in tandem: a reverse transcriptase domain and a primase polymerase, or PP domain. During a phage infection, the DRT7 enzyme synthesizes massive volumes of defensive DNA. It does this entirely without the presence of an RNA template strand. A reverse transcriptase operating without a template violates the established mechanics of enzymology. To sequence these nucleotides without a genetic guide, the enzyme requires a completely unknown method of molecular synthesis. To understand how this synthesis occurs, researchers shifted from theoretical models to empirical data by analyzing infected E. coli cells. They extracted the newly synthesized DNA produced exclusively by activated DRT7 enzymes during the infection window. Because standard sequencing struggles with highly repetitive DNA, they applied PacBio long-read sequencing technology to decode the exact biological output. This data scatter plot charts the lengths of the resulting DNA sequences, revealing a tight cluster isolated exactly at 1.8 kilobases. Furthermore, the sequence lacks genetic complexity, composed of greater than 60% alternating tracks of purely adenine and thymine. Researchers then applied structural alignment mapping to these repeating AT sequences to determine how they arrange themselves in three-dimensional space. The alternating blocks form a perfectly symmetrical palindromic hairpin. The sequences act as exact chemical mirrors, snapping together into a dense structure. Notably, this massive DNA hairpin remains covalently linked to the DRT7 protein that built it. To understand how a single enzyme rapidly constructs a 1.8 kilobase symmetrical hairpin without a genetic template, we must deduce the internal mechanics of DRT7 by working backward from the physical artifact itself, reverse engineering the machine. The investigation moves from the synthesized DNA product to a high-resolution structural map of the DRT7 enzyme prior to infection. Utilizing cryo-electron microscopy, researchers resolve the structure down to the angstrom level, allowing them to map the position of individual amino acids. In a healthy cell, DRT7 rests in an inactive state. It forms a symmetrical, closed dimer, with its two identical halves, called protomers, tightly locked together. The transition to an active state is triggered by a specific phage-encoded transcriptional regulator called Bas63-035. When the virus injects this protein, it inadvertently alerts the bacterial cell to the invasion. Contact with the viral protein triggers an asymmetric shift. Protomer 1 locks down as a rigid scaffold. Protomer 2 undergoes a violent 180-degree rotation, breaking the symmetry. This exposes two active sites, now chemically primed for synthesis. This strict structural lock-and-key mechanism ensures the lethal DNA synthesis protocol remains completely inaccessible until a viral threat actively forces the shift. We can now look directly into the newly exposed reverse transcriptase domain of protomer 2. Unlike a classical retroviral reverse transcriptase, this cavity is completely empty. There is no RNA strand present to guide the placement of the first nucleotide. Instead, DRT7 uses protein-templated initiation. The fixed physical geometry of the enzyme replaces the genetic code. The RT active site uses an arginine pocket. 4 rigid pillars map perfectly to a thymidine base. Other nucleotides physically clash and are ejected. The first thymidine anchors to residue Y682, feeding identical nucleotides to generate a 10-nucleotide poly-T tract. By substituting a nucleic acid template for an internal amino acid mold, DRT7 forces sequence-specific polymerization using nothing but its own physical structure. With the initial strand formed, the synthesis transitions to the second critical region of protomer 2, the primase polymerase, or PP domain. The poly-T tract releases from the RT pocket, shuttling across the complex to insert its free end into the PP active site. Inside, it acts as the initial primer and folds back 180 degrees to act as its own template. The internal architecture of the PP active site differs entirely from the RT site. It completely lacks the base-specific recognition residues of the arginine pocket. Without a structural mold, the PP domain relies entirely on Watson-Crick base pairing. It reads the folded poly-T template and selects its chemical complement, synthesizing a corresponding poly-A tract alongside it. This handoff results in a hybrid intermediate molecule, a chain containing perfectly alternating blocks of T and A. Generating a perfectly alternating palindrome requires two distinct enzyme domains operating under opposite selection rules, one dictating sequence structurally and the other chemically. A single handoff creates a short TA intermediate sequence. To bridge the mathematical gap to the massive 1.8 kilobase structure found in the bacterial cells, the enzyme employs a continuous feedback loop. The synthesis process utilizes an iterative handoff mechanism. The reaction does not terminate after one cycle. Instead, a reverse shuttle occurs. The newly formed poly-A chain detaches from the PP domain and is physically passed back to the RT domain. The RT domain resets the cycle, adding a fresh block of poly-T onto the returning chain. This initiates a rapid ping-pong repetition. The growing strand bounces between the RT and PP domains, adding alternating blocks with every pass. Older sequence segments continually fold back on themselves, acting as the required templates for the newly extending layers. This unchecked ping-pong mechanism carries a lethal macro-scale consequence. DRT7 rapidly generates a massive, rigid physical mass of DNA inside the bacterial cell. This suffocating mass of structural DNA physically jams the viral replication machinery. It executes the abortive infection, proving that reverse transcriptase's architecture.